The topic we'll be dealing today is about bacterial genetics and drug resistance in bacteria. What is genetics? Genetics is a science which deals with the heredity and variations. So it tells us how we resemble our parents and how different we are from our parents. So it is the science that helps us to know the inheritable characters from one generation to the other generation. Now coming to the bacteria, bacteria too follow this characteristic feature of inheritable characteristic. That is, they also show that characteristics are transferred from one generation to the other generation. That is, they also follow the law of genetics. So initially it was the Nageli who proposed the concept of pleomorphism, which states that only few bacteria shows variations, but later on with the development of pure culture techniques, it became apparent that different species of bacteria retain their characteristics from one generation to other generation, showing less changes or variation potential. So this concept was called as pleomorphism, and with the help of pure culture techniques, we are able to know that different bacteria can be classified into different species based on a particular character, and these characters are retained from one generation to the other generation. So this gave us a new science which is called as genetics, bacterial genetics, that helps us to get an insight into the genetic component of the organism and important characteristic features of this genetic component. That is, whether it is a DNA or RNA which acts as a genetic material, we are able to study in detail about the different characteristic features of the DNA. And with this particular bacterial genetics, we are able to get an insight into the other form of higher study that is called as molecular biology, that not only helps us to analyze the DNA but also manipulate or tailor the DNA according to our needs. So that is what is called as molecular biology. So in this particular topic, we will be dealing all about the genetic component of the bacteria. So first I'll start with the principles of molecular biology, where I'll be dealing with the central dogma, which is an important part of a bacterial genetics. So the central dogma is the one that tells us how DNA, while undergoing self-replication, also has the ability to produce the protein via the synthesis of mRNA, process of transcription and translation. So it is from the DNA, with the help of transcription, mRNA is produced, and the mRNA is the one which holds all the information for the synthesis of the protein, and it is a protein that usually carries out all the functions of a cell. And apart from protein, it is the enzymes that usually are helpful in carrying out the different biochemical activities. So DNA, while undergoing self-replication, also have the ability to produce protein via the two process which is called as transcription and translation. So it is the mRNA that holds all the information that are later translated into the proteins, which is an important component to carry out the biochemical cycles in the cell. So here we can say that DNA is a storehouse of the information of protein synthesis. So DNA is a polynucleotide chain, whereas protein is a polypeptide chain. Polypeptide is nothing but building blocks of amino acids. So the unit component of protein is the amino acid, and this amino acid is usually synthesized from the DNA, that is a polynucleotide chain. So before going with the protein synthesis, let us know what is a DNA and what is it usually made up of, through which the protein is usually synthesized. So coming to the structure of the DNA. DNA is deoxyribonucleic acid. So it is made up of three important components. One is a sugar component, the other one is a phosphate group, and the next one is an important nitrogenous group that are attached to the deoxyribose, that is a sugar. So it is a polynucleotide chain. That means there are many nucleotides that are present in a DNA molecule, and there are two nucleotide chains that are wound together in a form of a double helix. You can see the image. So they are usually wound around each other as a double helical structure, and the backbone of a DNA is made up of deoxyribose and a phosphate sugar. So to this deoxyribose is attached one of the four nitrogen bases that are called as adenine, guanine, thymine and cytosine. So among the four, adenine, guanine, thymine and cytosine, two are purines and two are pyrimidines. That is, one is adenine and guanines are purines, and thymine and cytosine are pyrimidines. So purines always pairs with the pyrimidine. So this type of pairing is called as complementary base pairing. So we have a backbone to which nitrogen base is attached. One of the nitrogen base is attached, and the two strands are wound together in a double helical form and stabilized by a hydrogen bridge that connects the two double helical structure. So adenine always pairs with thymine and guanine always pairs with cytosine. So there are equal number of adenine and thymine and guanine and cytosine. But the ratio of adenine and thymine to the guanine and cytosine varies from one species to the other species. This helps us to know the different organisms and different types of DNA that are usually present in an organism. So the component of purines and pyrimidines are always equal, but the ratio varies from one organism to the other organism. So the basic structure of the DNA is, it is a polynucleotide chain having a backbone and the 4 nitrogen bases. Now next is how this DNA usually replicates. So replication of DNA is nothing but synthesis of the copies of the DNA, and this DNA will be like transferred from one generation to the other generation. So coming to the replication part of a DNA, the replication is called as semi-conservative mode of replication. So in this particular mode of replication, the two strands of the DNA first will unwind with the help of different enzymes. Now the two strands that have unwound, it is not the whole DNA that usually unwinds. It is a part of the DNA that first unwinds to form a replicating fork on which a new daughter strand will be synthesized. So this parental strand that has unwound and found replication fork are called as template strand. Template strands are nothing but the master copy which dictates the sequences to the daughter DNA molecules. That's why it is called as a master copy. So one strand dictating the sequence to the daughter strand. So here, as I told you, the base pairing is completely complementary. That is, wherever there is A, the immediate next strand will have a T. So that is what is complementary base pairing. Wherever there is a G, there will be a complementary C that will be attached. So once the DNA unwounds at one end, there is a replicating fork that is formed. Each strand will behave as a template strand or a master copy on which a new daughter strand will be synthesized. So because the daughter strand contains one of the parental strand in its double helix, it is called as semi-conservative mode of replication. So it is not the whole DNA that first unwinds. It is a part of the DNA that keeps on unwinding, and new daughter strands will be keep on synthesizing successively. So it is called as semi-conservative mode of replication. So here we have the different types of enzymes that are involved to unwind the DNA, and we have an important enzyme to synthesize the daughter strand that is called as DNA polymerase. So DNA polymerase is an enzyme that keeps on adding the nucleotides depending upon the complementary base pairing. And whatever the nucleotides that are coming are from the genetic pool or the nucleotide pool that is present in the cell. So depending upon the particular base on a parental strand, the daughter strand will be successively synthesized. So this is what is called as semi-conservative mode of replication. Next, we'll be dealing about the differences between DNA and RNA. So it is in most of the cases DNA that is one that is acting as a genetic material, but there are organisms where RNA acts as a genetic material. So in this case, RNA will take the whole responsibility of carrying the genetic information from one cell to the other cell. So it is said that it is always true that DNA and RNA together cannot act as a genetic material. It is either a DNA or RNA that will act as a genetic material. So in case of the organism which has RNA as a genetic material, DNA will not be present. So coming to the similarities and the differences between the DNA and RNA, RNA is nothing but a copy of a DNA. So it has been synthesized from the DNA. But the differences over here is there is a difference in the sugar that is present. So here in case of DNA, it is a deoxyribose sugar that is present, whereas in case of RNA, it is a ribose sugar. And coming to the nitrogen bases, in case of in place of thymine, which is present in DNA, it is uracil that is substituted in the RNA. So this is the two different differences that can be seen based on the DNA and RNA. But the RNA is always complementary to the DNA and it is synthesized from the DNA. In case of the organism where DNA is absent, RNA acts as a genetic material. So it is an exact copy of a DNA. So there are different classes of RNAs that we know. We can classify depending upon their function. One is called as the informational RNA and the other one is called as the structural or functional RNA. So informational RNA is the mRNA that has all the information related to the synthesis of the protein. So from the DNA, mRNA is produced, which is called as a messenger RNA, and this messenger RNA holds all the information that can be translated into the proteins via two functional RNAs which are called as tRNA and RNA. tRNA is transfer RNA that brings amino acid during the protein synthesis, and rRNA is called as a ribosomal RNA. It is a place or a machine where the proteins are usually synthesized by a process which is called as translation. Now the difference between the prokaryotic protein synthesis and eukaryotic protein synthesis. Prokaryotic protein synthesis is very simple because the organisms are unicellular in nature. There is no complexity involved in the synthesis of the proteins, because in the prokaryotic organism the nucleus is not present. It is the nucleolus which holds the DNA, and the mRNA that is from the DNA is a direct transcript that can be involved in the protein synthesis. But in case of eukaryotic protein synthesis, there is a particular nucleus where the DNA is present. So first the DNA has to be copied onto the primary transcript, which is later processed to synthesize the mRNA. mRNA moves from the nucleus to the cytoplasm where the proteins are usually synthesized from the ribosome. And moreover, the DNA of prokaryote is different from the eukaryotes. Prokaryotic DNA is simple, whereas eukaryotic DNA contains two segments. One is called as the introns and the exons. So introns are intrusion elements. They are non-coding sequences, whereas exons are coding sequences. So that is the requirement for the processing in the eukaryotic protein synthesis, where the DNA is first translated or it is transcribed to the primary transcript, which contains both introns and exons. There is a splicing of introns where the introns or the non-coding sequences are removed, remaining with only the exon, that is the coding sequences. Now this is called as a primary transcript, which is later modified into mRNA by adding a 5' modification and a 3' modification. That is, at 5' they add a 7-methyl guanosine cap, and at 3' they add a poly A tail. Now this is an mRNA or the messenger RNA that usually transfers itself into the cytoplasm where the ribosomes are present and translated into the proteins. So prokaryotic protein synthesis is different from the eukaryotic protein synthesis, and the DNA of prokaryotes is simple, whereas the DNA of eukaryotes contains both coding and non-coding sequences, that is introns and exons. So hence the splicing is very essential to remove the non-coding sequences. So this is about the different types of RNAs. So one is the mRNA, messenger RNA, that is synthesized from DNA by a process which is called as transcription, has all the information for the synthesis of protein, whereas the other two RNAs are only at the functional level to synthesize the proteins. Now coming to the genetic code. Now what is a code? Code is nothing but it contains the information to synthesize the amino acid or add the amino acid during a protein synthesis. So this is called as a genetic code because all the information is present in the mRNA molecule, and this mRNA is nothing but a complementary copy of a DNA. So information from the DNA has to be translated into the proteins via the mRNA. So the mRNA has a code for the synthesis of protein, and this code is in the form of codon, which are nothing but the 3 bases of mRNA act as a codon to synthesize a particular amino acid. Take for example, the 3 bases are nothing but AUG. AUG will code for amino acid methionine. So this is the starting amino acid and it ends with a terminating amino acid. So the polypeptide is nothing but chain of amino acids. So polypeptide is functional only when it is a complete polypeptide that has undergone processing to take part in a cellular function. So the amino acids are synthesized from the 3 bases that are present on the mRNA which act as a codon. So we have different codons to synthesize different types of amino acid, and this genetic code has certain properties. So it is said that it is a triplet code and contains 3 bases. 3 bases codes for one amino acid. That means it is a triplet code, and this code is called as degenerate. Degenerate means a single amino acid can be coded by more than one codon. The triplet code synthesizes or adds one amino acid during the translation process, and the code is called as degenerate because a single amino acid can be coded by more than one codon, and we have 20 different types of amino acid for which we have different types of codons to synthesize a particular amino acid. So there is a single codon for only one amino acid, that is for methionine. AUG is the only one that adds an amino acid methionine during the protein synthesis, which is the starting codon in every organism. So ends with a terminating codons, and this codon or genetic code is non-overlapping. It means that once the 3 bases have been adding one amino acid, next immediate 3 bases will add another amino acid. So you don't have any punctuation marks in between to separate one codon from the other codon, and there is no overlapping between the codons. So first 3 bases, once they are coded, next immediate 3 bases will act for adding another amino acid. And always a polypeptide starts with an initiating amino acid and ends with a terminating amino acid. These terminating codons are called as nonsense codons because they don't code for any type of amino acid. So there are 3 such nonsense codons which is called as UAA, UAG and UGA. They are also called as stop codons. Once these stop codons are reached in an mRNA molecule, the synthesis of polypeptide completely stops, and this polypeptide will undergo processing to synthesize a functional protein molecule. So the genetic code is a code that helps us to know about the different codons that are present in an mRNA molecule that is required to add a particular amino acid during a protein synthesis. So this code is very important to get the information for the synthesis of polypeptides from the polynucleotide chain. So via the translation process. So synthesis of mRNA from DNA is called as transcription, and from the mRNA the code is translated into the proteins for a particular. So there are different genes that are present that synthesizes different proteins in a DNA molecule. So next important thing that we'll be studying over here is about the gene. Gene is nothing but a segment of a DNA that specifies a particular polypeptide. So there are innumerable number of genes that are present in a DNA molecule. It is not the whole DNA molecule that acts as a gene, but there are certain parts of a DNA molecule that specifies a particular protein. So that is called as a gene. So specifying a particular polypeptide or a protein, and it is made from nucleotides because it is a complementary copy of a DNA. It is usually the genes are nothing but are of nucleotides. That is, they contain all the 4 bases adenine, guanine, thymine and cytosine, which are translated into the transcript into the mRNA, which are later then translated into the proteins. And here the bacterial chromosome, coming to the bacterial chromosome, it is a double stranded circular form which is about 1000 micrometer. When we open it up, the length of which is 1000 micrometer, and we usually express in the form of kilobases, and it is the kilobases, that is a unit that represents the nucleotides that are usually present. So in case of bacterial chromosome, it is a 4000 kilobase pairs, and in humans it will be in millions. Around 3 millions of kilobases are usually present in the human genome. And the differences, as I told you, between the prokaryotic DNA and the eukaryotic DNA is the presence of introns and exons. So there is a difference in the processing in a case of prokaryotic translation and eukaryotic translation. So it has to undergo processing by removing the introns in case of eukaryotes, whereas there is no such need for to eliminate any type of introns that are present in the DNA. So DNA of prokaryotes does not have any introns, whereas eukaryotic DNA has introns and exon, which are called as non-coding sequences and coding sequences. So this is about the gene and the genetic code that holds the information for the synthesis of the protein. So in a bacterial cell, apart from the normal chromosome, we also have certain extra chromosomal genetic materials which are called as plasmids. So plasmids are self-replicating or autonomously replicating particles or molecules that are present in a DNA molecule, and this plasmids, though, do not confer certain properties to the bacterial cell, but they have certain important characteristic features that they can give to the host. So the presence or absence of plasmid will not affect the host to a much extent, but there are certain properties that are present on the plasmid that can be conferred upon to the host, and these characteristic features are nothing but the resistance and toxigenicity. Resistance towards different types of drugs, and toxigenicity is nothing but they produce, these plasmid produce certain types of antibiotic compound that inhibits the other closely related organisms. So they are autonomously replicating, that means they have their own origin of replication. So they replicate as and when the bacterial chromosome replicates, and they get transferred from one cell to the other organism. And during this process of transferring, the plasmid can also transfer the gene from one cell to the other cell, and because of this characteristic feature, we can make use of the plasmids as vectors in genetic engineering. Vectors are nothing but the cloning vehicles that carries our gene of interest in genetic engineering. And sometimes these plasmids also have the ability to get integrated into the bacterial chromosome. So this plasmid that has integrated into the bacterial chromosome is called as episomes. So the important characteristic feature of this episome is, when this episome is coming out of the bacterial chromosome, sometimes it can carry certain genes that are present in the bacterial chromosome and can transfer it to the other cell in whichever it is going to get transferred. So episomes are plasmid getting integrated into the bacterial chromosome. And you can classify the plasmids. There are different types of plasmids that are present in a cell. So classify the plasmids based upon their characteristic feature, that is whether they can transfer themselves on their own, called a self-transmissible plasmid or conjugating plasmid, or they can be non-conjugating plasmids. Apart from that, the other properties that they usually confer to the host, you can classify them based upon that, that is whether they are sexual plasmids that can transfer the genes from one cell to the other, or they can be drug resistant plasmid, because as I told, this plasmid contains certain types of gene that confers drug resistant to the host. So depending on that, you can classify the plasmids. And then apart from that, there is an important classification which is called as incompatibility grouping. Here the closely related plasmid cannot stay in a single bacterial cell, so they cannot stably exist or coexist in a cell. So they are different type of plasmid that can coexist, but not the same related or closely related plasmid. So this is called as incompatibility typing, incompatibility plasmid. So closely related plasmid cannot coexist in a cell and they cannot be stable, whereas unrelated plasmid can coexist in a cell. So this is called as incompatibility classification. And the most important characteristic features of this plasmid is they confer certain properties to the host to survive in a particular environment. Next topic in bacterial genetic is genotypic and phenotypic variation. Genotype is nothing but sum total number of genes that are present in an organism is a genotype. That is the genetic potential of a cell. The complete genetic character of a cell is nothing but a genotype. So genome is nothing but a sum total of a gene that is present in an organism, and it is the heredity constituent of an organism. Now coming to the phenotype, phenotype is nothing but the display or the expression of a genotypic character is called as phenotype, and it is completely based upon the environment in which that is to be expressed. Take for example tubercle bacilli usually is flagellated. It has flagella when it is grown in a broth, but when they are grown in a phenol agar, they do not produce their flagella. So that is the environmental influence that usually shows whether a particular component has to be expressed or not. Though the genes that are present are already there in a cell, but they depending upon the expression on the environmental condition, whether they are to be expressed or not, that is what is called as phenotypic variation. That is, there is a variation. They changes their character depending upon the environmental conditions. So these phenotypic variations, the other example I can give over here is the production of an enzyme which is called as beta-galactosidase, produced by an E. coli organism. So the genes for the production of this beta-galactosidase are present in a DNA of E. coli, but the translation of this protein, that is the synthesis of the enzyme, depending on whether the substrate has lactose in it or not. So if there is a lactose in the medium, the enzyme is produced. That is, there is an expression of a gene, and hence the enzyme to utilize the lactose is present. That is, lactose fermentation takes place. But when there is no lactose in the medium, the enzyme for the synthesis, that is beta-galactosidase, is not present. So again, there is an environmental influence over here. So the physical properties or the phenotypic characters of a particular organism varies depending upon the environmental condition, whereas genotype or genotypic variation usually does not take place. They are usually stable in nature, whereas the phenotypic variations are only temporary. That is, return of condition, again the organism becomes flagellated. So that is a temporary variation, and they are limited in their range, and they cannot be inheritable. They cannot be given to successive generation. That's why they are called as variation. Variations are not inheritable. They cannot be inherited to the next successive generation. Whereas when there is a change in the genotypic character, they are usually inherited. And sometimes this inheritable character in a genotype can be beneficial to the organism, or it can be lethal to the organism. So genotypic variations are usually stable in nature, and this genotypic variation takes place when there is a mutation in the organism. Whereas phenotypic variations are temporary. Only depending upon the environmental influence, you can see the change in the characteristic of a particular organism. So the genotypic and phenotypic variations that are usually brought about are mostly because of a phenomena which is called as mutation. So phenotypic variations are only temporary, whereas genotypic variations are stable. This is because of the mutation. Now let us see what are mutations. Mutations are nothing but they are random, undirected, heritable variation. So what I mean to say random means it can occur in any life cycle of a cell. They can occur in a vegetative cell, or they can occur in a somatic cell, or they can occur in any stage of a cell, whether it is a actively reproducing cell or it is a dormant cell. So it is a random event, can occur in any part of a life cycle and in any organism, and they are undirected. There is no direction for this mutation. That is, they can cause a wild type to form a mutant type, or from mutant type they can again get back to the wild type. So there is no particular direction for the mutation to take place, and they are heritable variation. As I told you, the difference between the phenotypic variations and the genotype is phenotypic are temporary, whereas genotypic they are stable one. They can be inherited from one generation to the other generation. So that is why it is called as genotypic variation. And this mutation involves a segment of a DNA. A segment means it can involve a particular base in a DNA. So when it affects a particular base in a DNA, it can either leads to the addition, deletion, or substitution of a base in a DNA molecule. So in any of the case, there is a change in the characteristic of a DNA, or the type of amino acid that has been synthesized based upon the three bases. As I told you, AUG codes for methionine, that is a starting. So if there is a change in the base due to mutation, either attacking the A or the U or the G, a complete change of amino acid will be replaced. So no polypeptide can be synthesized if there is no starting amino acid. So this type of mutation that occurs in a DNA molecule at a particular base is called as point mutation. So it lead to addition, deletion, or substitution of a base. And if such large number of point mutations are there in a DNA molecule, it can lead to chromosomal damage, ultimately leading to the lethal effects to the bacteria, killing the bacteria. So if there are point mutation in a DNA molecule, there is a chance that the bacteria can survive. But if there are chromosomal mutations in a DNA molecule, there is no possibility for the bacteria to survive. So these mutations are a point mutation, and they can be classified as missense mutation. Missense mutation is the one when there is a change in the base. As I told you, AUG, if any base is changed, it will lead to the synthesis or addition of different amino acid. That is called as missense mutation. There is no, like the normal sense have been changed, and then it can lead to deletion. Deletion and addition leads to the decreasing of the polypeptide or increasing of the polypeptide chain. In both the cases, it is lethal to the microorganism because a functional protein cannot be synthesized. And apart from that, there can be transition and transversions mutation. Transition is change from purine to purine or pyrimidine to pyrimidine. In transversion mutation, there is a change from purine to pyrimidine and pyrimidine to purine. So in both the cases, it is a transversion that is most lethal and brings about a lot of mutation to the organism. And apart from that, there is a lethal mutation. Lethal mutation is nothing but if the mutation is caused to in a organism that leads to the death of the bacteria, it is called as lethal mutation. And one of the example of this is called as a conditional mutation, where the organisms usually surviving in a normal range of 39 degrees centigrade. If this organism has been mutated by a conditional mutant, they can survive only at 35 degrees centigrade. So increasing the temperature will ultimately kill the organism. So conditional mutation usually are expressed during a particular environmental condition. So this is how you can classify the mutations into different types, and these mutations are usually caused in the organism due to two reasons. One is called as a spontaneous mutation, that is a natural mutation directed under the natural forces, and the other one is called as the induced mutation, where the human uses certain types of chemical agents to induce mutation in a DNA molecule. So in a spontaneous mutation, natural mutations, the mutation usually takes place during the replication errors. So sometimes there is a possibility that there might creep in an error during a replication process. Wrong base might be added in the DNA molecule that will lead to the mutant type of DNA. Okay, and in the case of induced mutation, we are inducing the mutation by making use of certain types of mutagens like chemicals, etc. So example of this chemical mutagens are fibromouracil, iodouracil, and we have acridine dyes, proflavin, agriflavin, etc. And the reason for the natural mutations are, as I told you, because of the replication of errors. And here the bases might undergo tautomeric shift, that is one change in the base configuration can add a wrong base in the daughter DNA molecule. So that is leading to the mutation in a DNA molecule. And here the mutation rates is very low. The frequency of mutation taking place in a normal or natural condition is very low, where it takes place to about 10 to the power of minus 4 to 10 to the power of minus 10 per bacterial division. So it is a very slow phenomena or very low frequency phenomena that can be seen in the environment. But when we are using induced mutagens to induce, we can alter the DNA molecule based upon our interest. And here we have, as told you, this mutation usually gives certain property to the organism which is called as survival advantage. Take for example, the organism is resistant to streptomycin. It can survive in a patient who is on treatment with streptomycin. So on a patient who is on a particular antibiotic, this mutation or mutated organism confers a survival percentage in the human host, where they completely multiply in spite of the drugs that are taken by the human to treat a particular infection. So it confers certain survival advantages for the organism to survive in that particular environment. And this phenomena was initially called as adaptation, that is change in the characteristic feature of the bacteria depending upon the environmental influence. But later on it has been discarded. But it is said that these mutations usually confers certain properties to the organism to survive in the particular environment. So you can classify again the mutations depending upon the nutritional requirement, the biochemical reactions and antigenic structures, etc. And the practical importance of this mutations is that they confer drug resistance to the organism, and they can be used in the production of vaccines. As I told you about the induced mutagens, we can use this to change the antigenic character of that particular organism to prepare the vaccines. Now coming to the recombination in bacteria, how this recombination or the transfer of genetic information takes place in an organism, and how it is inherited from one cell to the other cell. So this is called as recombination, bacterial recombination. Recombination is nothing but combination of a gene. So as already discussed in the previous slides, that these organisms show little variations. So they keep on giving the characteristic from one generation to the other generation with very low frequency of variation. But sometimes in nature there can take a recombination process where a combination of two genes will take place to produce a new type of a DNA molecule. So bacteria can also carry three types of recombination process, which are called as transformation, transduction and conjugation. So first I will be dealing about transformation. Transformation is nothing but transfer of genetic material from one cell to the other cell, and the agent over here is a free DNA molecule. So we can make use of free DNA molecule to transfer a gene of interest in a bacterial cell. So transfer of the genetic material can take place in the form of a free DNA, where the host organism can take up a free DNA from the surrounding environment. And this process of transformation was first given to us by a scientist called as Griffith. Griffith was working with a organism called as Diplococcus pneumonia. He was working with the pathogenic character of this organism that was highly virulent and was causing pneumococcal infection. So while studying on this particular organism, he has seen two type of strains of Diplococcus. One is called as a rough strain, that is 2R type of strain, and the second one is called as the smooth strains, that are called as 3S strains. So when he injected the experimental mice with 2R strain, he could see that the disease was not produced in the mice. So when later he injected the mice with the 3S strain, he could see that the mice died because of the pathogenic character of this 3S strain. Now next what he did was he heat killed the 3S strain and he injected into the mice, and he could see the mice did not die. And next step what he did was he made a mixture of 2R strains and the heat killed 3S strain and injected into the mice, and to his observation he saw that the mice died. When he isolated the organism from the dead mice, he could see the organisms of 3S type. But when he injected it, it was 2R plus 3S strains, heat kill 3S strain. So the organism that was isolated was 3S type. So he confirmed that there is certain characteristic feature that is present in heat killed 3S strains that has been transferred to the 2R to change into 3S type. So there is a transforming principle that has been transformed from 3S, heat killed 3S strains, to the 2R strain to change the 2R strains to the 3S strain. So this, he said that there could be some transforming principle that has been transformed. Now later this was further, the experiments were carried out, and finally three scientists called as Avery, MacLeod and McCarthy finally proved that transforming principle is nothing but the DNA molecule that has been transformed from 3S, heat kill 3S to 2R and change the 2R strain to the 3S strain. So here the DNA has been transformed from the 3S strain, heat kills 3S strain, to the 2R strain. Later on, further the experimental works were carried out by Hershey and Chase using bacteriophages, and he said that it is the DNA that acts as a genetic material and not the proteins. So finally it was confirmed that DNA is a genetic material, and this DNA can be transferred from one generation to the other generation and one cell to the other cell, taking part in recombination. So this is one first part of recombination, where certain bacteria can show a recombination of the bacterial genes. Combination of two different types of genes can take place by the transformation. And next is transduction. Transduction is nothing but the recombination mediated by bacteriophages. Bacteriophages are the viruses that infects the bacteria, and the stages that are involved in the infection of the bacteria with the viruses is that first it attaches to the bacteria and it releases the nucleic acid into the host, and this nucleic acid takes over the host mechanism, degrades the bacterial chromosome, and the viral nucleic acid takes over the host mechanism and synthesizes progeny viruses. And the progeny viruses are usually synthesized in different particles. The head is synthesized separately, the tail is synthesized separately, the nucleic acid is synthesized separately. So all these will assemble together to produce a complete progeny molecule, and similar such progeny molecules are produced, and ultimately they lyse the bacteria and then they are getting released to infect the other bacteria. So this is the life cycle how it usually takes place in the bacteriophages. Now during transduction, it so happens that during the packaging, that is during assembly, there is a possibility that some of the bacterial chromosome might creep into the phage head. In spite of the in place of the viral genome, bacterial genome might get infected, or it can creep in. It is called as a packaging error, and the progeny virus will have some part of the bacterial chromosome. Now this phase, when it infects the other organism, they can transfer the bacterial gene. So this is one way of carrying out the recombination process with the help of bacteriophages, which is called as transduction. Now this transduction can again be of two types. It can be generalized transduction or specialized transduction. Generalized transduction means any part of the bacterial chromosome can be transferred, whereas specialized transduction usually takes place in a life cycle of a bacteriophage, which is called as lysogenic cycle. So bacteriophage has two types of life cycle. One is called as a lytic cycle, the other one is called as a lysogenic. So previously that I have this said about is a lytic cycle. So lysogenic cycle is nothing but once the viral nucleic acid enters into the host cell, the viral nucleic acid gets integrated into the bacterial chromosome, and it lives in harmony with the bacterial chromosome and multiplies as and when the bacterial chromosome is multiplying. So this is called as lysogenized bacteria, and the DNA of the virus usually getting incorporated into the host bacteria is called as a prophage. Now this prophage while coming out from the bacterial chromosome usually can take a part of the bacterial chromosome. So when it is taking a part of the bacterial chromosome, it is called as a specialized transduction, because while getting incorporated in the bacterial chromosome, they get incorporated between two genes, which is called as gal and bio genes. So while coming out of that particular the prophage coming out from the bacterial chromosome, it sometimes happens that it can take away the gal gene or the bio gene and then give it to the other bacteria when the virus infects the other bacteria. So this is a specialized transduction where specific genes from the bacterial chromosome are taken up by the prophage, and the lysogenized lytic cycle, when it is taking place, it is a generalized transduction. Any part of the bacterial chromosome can be transferred to the recipient bacteria or the bacteria which is infected by this particular type of bacteriophages. So example of a specific bacteriophage is a lambda phage of E. coli that can transfer specific genes called as gal or bio genes during its transduction. And sometimes not only the bacterial chromosome, but the bacteriophage can also take plasmids and episomes from the bacterial chromosome and can transfer it to the other cell. And this bacteriophage is mostly used in genetic mapping in recombinant DNA technology to know where the genes are usually present in a DNA of a host. Now coming to the lysogenic conversion, it is also a part of the bacteriophage, part of life cycle, as I told you, lytic cycle or lysogenic cycle. In lysogenic cycle, they get integrated into the bacterial chromosome, and it is called as a prophage, and the bacteria is called as lysogenized bacteria. And this lysogenized bacteria usually they bring about lysogenic conversions. They make the host susceptible to bacteriophages, and they prevent the bacteria or the virus from immunity or super infections. And apart from that, these lysogenized bacteria, the DNA of the virus has certain properties or certain genes that confers toxigenicity to the host cell. Take for example, in the diphtheria bacteria, The bacteria as self is not toxinogenic. But when it is lysogenized with the viruses, it is the toxin gene that is present in the viruses that makes the bacteria toxinogenic bacteria. So they, apart from making it susceptible to the bacteriophages and preventing from super infections, this lysogenic characteristic feature of the viruses also confers certain drug resistance or toxinogenic property to the host cell. So the best example is the diphtheria bacilli. And once the virus nucleic acid is removed, the organism is not able to cause any type of infection, because there is no tox gene that is present in the bacteria to cause infection or cause pathogenesis to the human host. Next is conjugation. So three type of recombination. First one we have dealt about transformation, where the agent is a free DNA that can be taken up by the host cell. Next is a transduction, which is mediated by the bacteriophage, and the last one is called as a conjugation. This was proposed by Liederberg and Totem, and this particular conjugation process requires cell to cell contact, and it is in few organisms that has these plasmids, which are called as F plasmids, that are able to produce a conjugation tube that can bring the two cells together. That is, one is a donor cell, the other one is a recipient cell, and through the conjugation tube the plasmid can transfer the gene from one cell to the other cell. So it is not only the transfer of plasmid genes, but sometimes the plasmid, when gets integrated into the host bacterium, can take bacterial genes also, and it can give it to the recipient cell. So here the most important characteristic feature of conjugation is the formation of a sex pilus produced by only organisms that have F plasmid, and mostly seen in gram-negative organism, and it is also called as a fertility factor. So the fertility factor, depending upon it, presence or absent, the cell is called as F plus cell or F minus cell. F plus cell is a male cell or a donor cell. F minus cell is a female cell or a recipient cell. And once the transfer of genetic information takes place, the F minus cell also becomes a F plus cell and can transfer the genes to the other cell. And another important component in F factor is the HFR factor. HFR is nothing but high frequency recombinant factor. HFR strains are usually formed when the plasmid DNA gets integrated into the bacterial chromosome. So this is called as an HFR strain. So high frequency recombinant strain. Now the HFR strain also has the ability to produce a sex pilus and transfer the genetic information. But complete transfer of bacterial chromosome will not take place. So it is called as sexduction, and incomplete transfer does not lead to F minus strain getting converted to F plus or HFR strain. F minus will always remain F minus only, because complete transfer of genetic information is not taking place. Now F prime is another important factor of F, where the HFR strain, the fertility factor or the plasmid, when it has got integrated into the bacterial chromosome, it is called as HFR. But while coming out, if the plasmid takes some part of the bacterial chromosome, leaving behind its DNA molecule in the bacterial chromosome, this plasmid is called as F prime plasmid. So F prime plasmid also has the ability to transfer the bacterial gene from one cell to the other cell. So there are F factors. There are different types of F factor. F positive is male factor, F negative is female. When there is a conjugation taking place between two, the F minus gets converted to F plus. And we have HFR, where the plasmid gets integrated in the bacterial chromosome. This is called as high frequency recombinant plasmid or high frequency recombinant bacteria. Now when the plasmid comes out of the bacterial chromosome taking some part of the bacterial gene, it is called as the F prime plasmid. Apart from this plasmid transfer, there is also another factor that is transferred from one cell to the other cell, leading to recombination, which is called as cholicinogenic factor. So some bacteria have these plasmids, which are called as cold plasmids, that synthesizes certain types of colicins or antibiotics that inhibits the closely related group of organisms. So in order to survive in a particular environment, these bacteria produce colicins that inhibit the growth of other closely related bacteria. So this cold plasmid also have the ability to get transferred from one cell to the other cell. Now because there are different types of colicin produced not only by the gram-negative organism but also by the gram-positive organisms, the name is given as colicin, and it it is called as bacteriocins. Bacteriocins are nothing but colicin produced by different types of bacteria. Take for example, we have Pseudomonas pyoceanae. They produces pyocin, and diphtherycin produced by Corynebacterium diphtheria. So these organisms these bacteriocins are not only present in a gram-negative organism but can be seen in gram-positive organism, and hence the name is given as bacteriocin. And they also have the ability to transfer genes from one cell to the other, and they are called as transferable, episomal and infectious and drug resistance plasmids. And the last type of plasmid that is very important in medical field is the RTF, or the resistant transfer factor, or R plasmid. R plasmid is the one that confers multiple drug resistance in an organism. So as we know that the plasmid usually confers drug resistance in an organism. So now we have seen that different organism has developed drug resistance not against 1 or 2 antibiotics but more than 8 different types of antibiotics. So they are called as R plasmids. So R plasmid is a combination of 2 plasmid and 2 important factors. One is called as a resistant transfer factor and the other one is an R determinant. So this type of plasmids was first seen in the outbreak of Shigella dysentery in Japanese. So Japanese scientists have discovered that the patient who are suffering from this Shigella usually excreted certain types of bacteria, Shigella and E. coli, that have multiple drug resistance, and they have seen that the drug resistant factor is being transferred from one cell to the other cell. It is not only between the closely related species but also in the different species. That is, Shigella was able to transfer this multiple drug resistant factor not only to its closely related species but also to the E. coli. So this becomes a dangerous situation when we are on antibiotics, because they do not cause any type of harm to the organism, and this organism will survive in a patient who is on antibiotics with this particular type of antibiotics to treat a disease. So it has become a medical concern now to eliminate these type of organisms which are multiple drug resistant. So RTF is a resistant transfer factor along with the R determinant, and it is the R determinant that has the resistance towards the different types of antibiotics. So it can be more than 8 antibiotics to which a particular organism can be resistant towards different types of antibiotics. Sometimes this RTF or the R factor is a complex factor with 2 factors, that is RTF as well as R determinant. In some organism they can live separately, that is as an RTF plus R determinant. So in that case the R determinant cannot be transferred from one cell to the other cell. It is only the RTF that can be transferred. But when they are in complex complete plasmid RTF plus R determinant, they are able to transfer the genes to not only the closely related species but also to the other group of organisms. So bringing about a drug resistance towards a wide variety of antibiotics. Now the next important topic here that we'll be dealing about is the drug resistance in bacteria, which is of medical concern, important medical concern, because most of the bacteria are now becoming resistant towards different types of antibiotics, and it is becoming difficult for us to treat these infections with the antibiotics. Now the drug resistance in bacteria can be attributed to either the mutation or the gene transfer. So it is said that the reason for drug resistance in the bacteria can either be due to mutation or the gene transfer. So if it is mutation, as already told, it can be due to certain types of induced mutagens or the spontaneous mutation that can creep in during replication process, and that can make the organism resistant towards a particular type of antibiotic. And it is, if in case of gene transfer, it is mostly due to the conjugation process where the transfer of plasmid takes place from one cell to the other cell, and the plasmid carries the genes for the resistance. The biochemical mechanism of explanation that are given for the drug resistance in bacteria is they can carry an alternative metabolic pathway. Now there are certain antibiotics that targets metabolic pathway of an organism. So what this organism does is, they are so smart enough that they are completely changing the pathway by which they are producing a particular product during giving a metabolic pathway. So they choose an alternative pathway for the synthesis of a metabolic product. And apart from that, they can also produce certain types of enzyme to inactivate the antibiotic. Take for example penicillin. Penicillin is an antibiotic that is used to kill most of the organism. It is a broad spectrum antibiotic. Now the organism has developed resistance by producing an enzyme called as penicillinase that breaks away the beta-lactam ring of the penicillin, that is usually active upon the organism. So no longer the penicillin is active to act against the organism. So this is the most important part of the organism, that they can inactivate the antibiotic and hence become resistant towards a particular type of antibiotic. And apart from that, the organism can take up the antibiotic, but immediately they will pump it out from the cell, which is called as efflux pump technique in the organism. So bacteria are so smart enough to take part in all these activities to prevent itself from dying or survive in an antibiotic environment. And the mutational resistance is very important when it is concerned with Mycobacterium tuberculosis treatment. Now the tuberculosis treatment was initially given with the streptomycin, but the organism have become very resistant towards that particular antibiotic, that is streptomycin. So a combination of drug is usually given, so that if the one antibiotic, if they have developed resistance, the other antibiotic is active towards that particular organism. So here the organism is not only developing resistance towards different types of antibiotics, that is called as multiple drug resistance factors in the organism. So the treatment is nowadays that is given for the tuberculosis is to change the pattern of antibiotics for every 6 months or 3 months. So there is a combination of drugs given to inhibit the like multiple drug resistant organism and to inactivate this organism and kill the organism. So it is a combination therapy, you can say. A combination of different drugs is given to eliminate this multiple drug resistant organism. One thing that is to be kept in mind is the frequent usage of antibiotics can also lead to the resistance in the bacteria. So it has to be kept in mind or borne in mind while taking the antibiotics to not make this organism resistant towards that particular antibiotic. And complete course of antibiotic should be done to eliminate the emergence of this drug resistance organism. And here transferable drug resistant factors is another important thing, that is the RTF and R determinant that usually causes the drug resistance in the organism. And the R factors usually produces enzyme to inactivate the antibiotic, and hence there is an emergence of drug resistance in bacteria. And use of these antibiotics in animal feeds can also lead to the drug resistance, and these drug resistant bacteria can be transferred to the human when they consume the animal as their nutrition. So the use of antibiotic also should be eliminated, and the government has taken steps to limit the use of antibiotics in the animal feed and veterinary actions. Now apart from this, the plasmids which are acting as extra chromosomal material, there are certain DNA, genetically discrete segments of DNA that have the ability to move from one part of the chromosome to the other part and from chromosome to the plasmid. So these are called as jumping genes, nothing but the transposons. So transposons are nothing but the segments of DNA which are genetically discrete and can move from one part of the chromosome to the other part by a mechanism which is called as cut and paste mechanism. Sometimes they can also do a copy paste mechanism, but mostly it is a cut and paste mechanism where the segment of the DNA is cut from one part and it has been introduced into the other part. So they are called as jumping genes. And how are you going to identify these jumping genes in an organism or in a DNA molecule is the presence of the inverted repeats. And the existence of these transposons was first given us to us by a scientist who is called as Barbara McClintock when she was working with the modeling of kernels in the maize. So she was the one who is given as a concept of this particular genetic segments of a DNA that are present that can relocate from one part of the chromosome to the other part. So they are called as transposon. And and the simple type of transposons are called as IS elements. They can be recognized by the inverted repeats. So these inverted repeats are usually flanking a single gene or more than one gene that can be present, and for the integration of this transposon, there is no requirement of genetic homology. Usually when there is a homologous segment, then the DNA can get integrated into the chromosome, but here in case of transposon, there is no such need of any genetic homology that is required to integrate. So it is the transposon that adds a bulk to the DNA molecule without any genetic homology, and they can also amplify the genetic transfer. So apart from that, the genes that are present in the transposon also can carry certain types of drug resistance. So when this type of transposon gets integrated into the bacteria, they make the organism drug resistance towards a particular antibiotic or a drug. So this is all about the classical genetics that we have dealt, and the component of the genetics that is usually seen in bacteria. That is, we have seen what is a DNA molecule and how it replicates, what is a gene, what is a genetic code, and how the proteins are usually synthesized from the DNA molecule. And apart from that, we have seen about the extra chromosomal material, that is the plasmids that are usually important to confer certain properties to the organism. And then we have seen how the recombination takes place. So bacteria usually reproduce by binary fission, but sometimes recombination also takes place to combine two different types of gene to produce a new variety. And the recombination process is transformation, transduction and conjugation. And next topic that we'll be dealing about is the next level of molecular genetics, that is called as molecular genetics or molecular biology. So an insight into the microbial genetics helps us to develop a new science which is called as molecular biology, that deals with the analysis of the DNA and manipulation of the DNA or tailoring of the DNA according to the human needs. So here we can manipulate the DNA and analyze the DNA and use this in various fields of molecular biology. And one of the most important application of molecular biology is the genetic engineering or recombinant DNA technology. So here we are carrying out technique to produce a new type of a DNA by combining two different types of DNA. Or in this case of recombinant DNA technology, we can insert our gene of interest in the microorganism to amplify our gene of interest and to get a functional product from our gene of interest. So the steps that are involved in recombinant DNA technology is we require a donor DNA molecule which contains our gene of interest. We require a cloning vehicle which is called as a vector. We require different types of enzymes that are used in recombinant DNA technology. And one of the important part of the recombinant DNA technology is restriction endonucleases, the enzyme which are called as molecular scissors. And finally, we need a recipient cell where we can transform our gene of interest and produce our gene of interest, or amplify or produce a product from our gene of interest. So there we have four different components that are required for genetic engineering. Isolate the donor DNA and cut it with the help of restriction endonucleases into variable lengths, and our gene of interest is selected, and then it is cloned and it is transferred into a cloning vehicle which is a vector. Vector can be a plasmid, which is an autonomously replicating unit which is present in the bacteria, or it can be a bacteriophage, and this will take your gene of interest, and hence it's called as cloning vehicle. Now this cloning vehicle, once it has been integrated with our gene of interest, it is called as a recombinant DNA. Now this recombinant DNA is transferred into the bacterial cell. Now why are we selecting the bacterial cell over here is because the generation time of bacteria is very short, and we can amplify a gene of interest at very high rates when compared to the eukaryotic cell. And apart from that, the bacteria are very simple in structure, and we can get our product very easily. When the organisms are growing, they release a product into the environment or into the media. So it is easy for us to collect that particular product from the organism. So first the step is isolate, then cut it with the help of restriction endonuclease, integrate into the vector to form a recombinant DNA and transform into the host cell, that is the bacterial cell, and ultimately amplify or produce your product. So one of the important discoveries that helps us to know about the molecular biology is the identification of restriction endonuclease. So these enzymes are usually produced in bacteria to protect themselves from the foreign DNA. So this restriction endonuclease was used now to cut DNA of variable fragments in recombinant DNA technology. So these restriction endonucleases usually cleaves the double stranded DNA molecule at certain sequences which are called as palindromic sequences. So they are specific oligonucleotide sequences that are present in a DNA molecule. And when they cut the DNA molecule, the same restriction endonuclease is used to cut the vector DNA molecule, where we can integrate our gene of interest and ligate it with the help of a molecular glue which is called as ligases. So there are different types of restriction endonucleases. Examples are EcoR1, Hint3, Tag1, etc. So for different types of integration, different types of cutting at different types of sequences, we can make use of the different types of restriction endonucleases. The main objective of restriction endonuclease is to digest DNA into fragments of variable length. So you can see where they cut at a particular sequence on the slide. So similar type of restriction endonuclease is used to cut the vector DNA molecule. So ultimately ligated. Now apart from that, the other thing that I used in molecular biology are called as DNA probes. DNA probes are nothing but single-stranded radioactively labeled complementary copy of a particular length of a DNA molecule. Usually it is about 20 to 25 nucleotides and mostly used to identify the antigen of the infectious diseases. Now we know that antigenic characters are nothing but they are usually of the DNA molecule. So take the DNA molecule of the organism and then denature it to produce a single stranded form. Add the DNA probes which if complementary they go and bind and hybridize to produce rays that can be photographed. So depending upon the complementary base pairing, you will know whether that particular infection is there in our body or not. So they are usually used in the serological techniques to identify the antigenic character of a particular organism. So it is easy identification. You do not take hours or the classical techniques which takes days together to identify the organism. That is, you do not need for the growth of the organism and all. Directly you can make use of a probe, take the DNA, hybridize it. If it is complementary, it will hybridize. If it is not complementary, then there is no hybridization. So it is easy identification of an infectious disease. Next is a blotting technique, another important technique to identify the homologous DNA present in a particular sample. So here we take the DNA, you have to digest it with the restriction endonuclease, run on a gel electrophoresis and then blot it on a nitrocellulose disc or a nylon sheet and then denature it to produce a single stranded, and to this you will add a single stranded radioactively labeled probe to produce a complement to make it hybridize and then identify with the help of X-ray photographs. So it is easy technique to identify whether a homologous DNA is present in a particular sample or not. So again, it does not take much time to identify the organisms or the DNA that is usually present in a particular sample. So when using DNA, it is called as Southern blotting technique. If the sample contains RNA and you are identifying the RNA, homologous RNA with the help of DNA probes, the RNA probes, then it is called as northern blotting. And if proteins are used to identify, it is called as western blotting, and if antibodies are used to identify, then it is called as immunoblotting. So immunoblotting is mostly used to identify the antibodies present in an HIV patients. So another important application of the molecular biology is the polymerase chain reaction. This is very important application of molecular biology, where you can amplify a small amount of DNA to a very large amount. So it helps in identifying the criminals who are usually involved in a particular criminal, or it can be used to identify for the paternity test, or to identify the or amplify the DNA molecule which is of very low quantity. So the most important application is polymerase chain reaction to amplify the specific DNA sequences, developed by Carey P. Mullis in 1983. So usually carried out in an automated system having different steps. So first here we have a denaturation step. We take the sample And then to this mixture we will add a tag polymerase, we will add a primer of short length, and then we add the nucleotides, and we usually carry out this in an automated system with different steps. First, there is a denaturation step that takes place at 94 to 96 degrees centigrade, where the denaturation of the DNA will take place. Denaturation is to separate the two strands of a DNA molecule. And then we have annealing temperature at around 68 degrees centigrade, where the primer will go and bind to the DNA molecule. And next is we have the elongation process, where the DNA polymerase or the DNA tag polymerase that is used to elongate the chain to form a complete DNA molecule from the primers. So the polymerase that we are using is called as a tag polymerase. So tag polymerase is the one that can withstand a very high temperature around 90 degrees centigrade. So usually isolated from an organism that can survive in a very high temperature. So this is a one round of replication, one round of application you can say. So similarly you can have 20 to 50 rounds of application to increase the amount of DNA molecule that can be used for the analysis and manipulation of a DNA molecule in molecular biology. Now, apart from that, the application of molecular biology is in the molecular epidemic and genetic mapping. So, molecular epidemic is you need to know which organism is usually causing an epidemic diseases in a particular area. So, you can identify the organism by plasmid profile analysis. You can analyze the plasmid, because plasmids are usually present in only few organisms, and depending upon the presence or absence of plasmid, you can classify the organism. And then you can go for genomic fingerprinting. You can go for PCR microbial isolate of epidemiology, like which organism is usually causing that particular epidemic can be easily known with the help of molecular epidemiology, and you can go for preventing this epidemiological diseases. Next thing, the wide application of molecular biology is the genetic mapping. So with the help of genetic mapping, you will know the genetic sequences that are present in an organism or the human. So the most important project that has been taken up is to know the complete human genome, a genetic makeup of a human being. So that is called as a human genome project. And here we can delineate the genomic sequences that are present in a particular DNA molecule. So this is a wide application of molecular biology that helps us to develop different products with the help of recombinant DNA technology.